Good afternoon, friends. This is Bhavana Suryawanshi, Assistant Professor from Department of Biotechnology. Today, we are going to see a new type of mutation, that is a base substitution mutation. Under this particular topic, we are going to see the types of base substitution mutation, followed by the mechanism of base substitution mutation with examples. Now, as we all know, mutation is a permanent change in DNA sequences. Okay, so mutations are classified under point mutation, insertion mutation, deletion mutation, duplication mutation, as well as inversion mutations. So base substitution mutation basically is coming under the type of point mutations. So let us see what is base substitution mutation. So when one nucleotide base is replaced by the another in a DNA sequence. So here students, the sequences are not getting added or deleted. Instead, a single nucleotide base is replaced by the other one. Okay, for example, if you can see the normal base sequence that is being shown here is A, T, G, C and T. This sequence is a normal, and when you see a mutated, when it is substituted by another base, that is A, C. Here T is replaced by C. Okay, so what could happen if such changes are occurring inside a DNA sequence? So it could have two consequences. It may have some effects, or they may not have any effect. Okay, so base substitution mutation are of different types. So first type of base substitution mutation is called as missense mutation. Second is silent mutation, and third is nonsense mutations. So what is a result of the substitution, we will see under different types. So first of all, let's start with silent mutation. So base change, whatever substitution that is occurring inside the sequence of a DNA is not altering any amino acid. Okay, like we know DNA is being transcribed into mRNA, and mRNA it is translated into protein, right? So even if a single base is changed, it is not changing the amino acid. Now students, do you know why it is so? Because it is redundancy of genetic code, because one or more codon can code for a single amino acid. For example, GAA, it is converted into GAC. Now GA is also one codon, right? And one base is substituted, so A is converted into G. But what is the net effect? Both of the codon is coding the same amino acid, that is glutamate. Okay, so here there is no net change. Even if there is a mutation, it is not having any effect. Okay, so no effect in protein function. Such example can be given as a beta globulin gene, wherein there is no change in the functions. Okay, so below is a figure of different amino acid that has been coded by more than one codon. Okay, coming to the next is missense mutation. Now missense, now if you try to split this term, miss is something which is not leading correctly, right? So base change that is altering one amino acid in the sequence. So whatever the amino acid a wild type sequence might have, it is being changed, it is misleaded, it is changed to some other one. For example, GAG, it is getting converted by GTG, wherein A is substituted by T. So GAG was a wild type. It was initially coding for glutamate, and it is converted into valine when GTG it is being changed. Now what is the result over here? Here you will have some drastic effect. One such rational example is sickle cell anemia. So here, if there is a missense mutation wherein glutamate is converted into valine, this is leading to change in the structure of hemoglobin. Here, if you can see in the picture, GAG was a normal codon. Okay, it was coding for a normal amino acid, and you can see in under the sickle cell condition that protein has altered the structure. and because we know structure and function of the proteins are interrelated, right? So the structure and the function of both this RBCs, this hemoglobin, because hemoglobin is a major carrier, right? Carrier of what? Iron and oxygen, right? So that has been converted into a different structure, that is sickle structure. You can clearly see the difference between the RBCs, normal RBC and sickle cell RBC. So this is one of the rational example of missense mutation. Now, third category is nonsense. Nonsense mutation means here the base is again changing, but here it is not changing to some other amino acid. Instead, that amino acid is converted into stop codon. Now we know what is a stop codon. Stop codon, if it is inserted into a growing polypeptide chain, it will terminate the protein synthesis. It will terminate the translation into a protein. For example, TAC, this is a normal codon, right? It is being converted into a stop codon, like for TAC, which was initially in a normal condition, it was coding for tyrosine. It is getting converted into TAG, that is a stop. Now here, why TAG? Because we know under mRNA doesn't have T, right? It has U. So UAG again is a stop codon. So what is the net effect of nonsense mutation? There is early termination of polypeptide chain. So whatever should be the normal length of the protein, that is not acquired, that is not achieved. Such proteins which are incompletely prepared, such polypeptides which are incomplete, are called as truncated proteins, because there was a nonsense mutation occurred in the due process, which has resulted in the truncated protein, incomplete protein. So such incomplete protein again may have effect. That effect could be abnormal effect, right? So one such example is Duchenne muscular dystrophy. So you can see in the picture, one is a normal muscle mass, right? And in the other one is the muscle mass. You can see there is a drastic reduction in the muscle mass. Now why the muscle mass is reduced in case of the second case? Because it has led to truncated protein. So whatever the muscle protein that is normally present and under wild type condition has converted into a mutated form, and that particular syndrome, it is called as Duchenne muscular dystrophy. Muscle weakness, the mass of the muscle is drastically reduced. That patient is not having that strength, right? So this could be a drastic change due to the insertion of stop codon. Okay, so moving further is, what is the molecular mechanism of this base substitution mutation, how it may cause. So they are caused by errors during DNA replication. So if we know, because DNA has been replicated with utmost speed, right? So there may be chances that during the replication a wrong base is incorporated. So this is coming under the errors that is occurring during DNA replication. Some chemical mutagens such as nitrous acid may also lead to base substitution mutation. Not only chemical, we also have physical mutagen, that is radiation. So which radiation can change it? Maybe X-rays, maybe UV rays. They can also lead to base substitution mutation. So again, if you can see, adenine is replaced by cytosine, that is change in the codon, and altered protein is observed. So molecular mechanism could be anything. It could be an intrinsic mechanism occurring within cell, no external moiety is involved, or it could be some mutagens. So mutagens can technically be chemical mutagen, or it could be physical mutagen. Now, what are the consequences of base substitution mutation? As we have already discussed, under the silent mutation we have seen, it is not causing any harm. So it could be beneficial, it could be harmless, or it could be harmful. Okay, so outcomes. So if the change is leading to a normal protein itself, that we can call that silent. If the protein is altered, that is missense. And if no protein, but is truncated protein is observed, that is nonsense mutation. So the total outcome of all this base substitution mutation can lead to genetic disorders, as we have seen the example of sickle cell anemia and Duchenne muscular dystrophy. Or if it is beneficial, some change is increasing the protein function, it is rendering the wild type gene more, strengthening the wild type gene, it is making that particular cell more adaptable, so it may also lead to some positive effects. So that positive effects could be evolutionary variation, so changing in the alleles, changing in the gene may be beneficial to the organism at higher hierarchy. Okay, so this is overall consequences of base substitution mutation. So this is the summary. As I said, DNA, there is a change in the bases, there is a transcribed codon that we can see in the screen, and wild type, silent mutation, missense mutation, nonsense mutation. This could be the three outcome of the mutational events that is occurring. Okay, so what we have seen, base substitution, this is very important, student. Here you need to understand that the nucleotide sequence is not been added or deleted, it is just replaced. So the total number of the polypeptide chain or the sequence is constant. So three types we have seen: silent, missense, nonsense. So changes may affect the protein, it may affect the structure of the protein, it may also affect the function of the protein, and it plays a key role in disease and evolutions. So these are the references. Thank you so much.